Okay. Um, well, thanks everyone who stuck it out here for our last session. I promise this is the last time you're going to hear from me today. I'm going to put on a slightly different hat and talk about a big collaborative effort through the Include project at NIH, of which I am one very small cog in a giant machine. And so I'm going to try to do justice to all of the wonderful collaborators that have contributed to this effort. And what I'm going to talk about today is an initiative called the Include Data Coordinating Center. So what the mission of this is to accelerate discoveries that continuously improve and enrich the lives of people with Down syndrome through accessible data resources that enable interdisciplinary collaboration, connected communities, and the expansion of a global Down syndrome knowledge ecosystem, which sounds like a lot of buzzwords, and that's because that's what it is. So I'm going to try to break this down a little bit and hopefully share a little bit more about what this means at a practical level. So what is the coordinating center? So NIH called for the creation of a coordinating center to accelerate the discovery of etiology and biologic pathways underlying the co-occurring conditions of Down syndrome by facilitating access to and querying of data from cohorts of individuals with Down syndrome. What does that mean? Quite simply, NIH had put a fair bit of money over the years into the development of cohort studies that generated a lot of different types of data, and I'll talk a little bit about what some of those look like, but they were siloed. They were living in different places. They weren't necessarily interoperable in such a way that they could talk to one another. And so the thinking was taking lessons from other examples of biology, which I'll touch on just very briefly as we move throughout the presentation. How do we accumulate all this data? How do we format all this data? How do we allow these data to talk to each other such that all of our various studies are informing all of the other studies and improving our ability to access those data and make discoveries using them? And so there's three prongs to this. There's the administrative and outreach core, which provides program management, outreach, and education. The data management core, which involves data collection, processing, and harmonization, and the data portal core. And this facilitates access to an analysis of data via an online web portal. So as I mentioned when I started, this is a huge team effort. These are just some of the team leaders. So the AOC, the administrative core, this is led by Dr. Joaquina Espinosa at the Crnic Institute. The DMC is a collaborative effort from Brian O'Connor of Sage Networks, Melissa Handel, who is at the Anschutz Medical Campus with us, and Carol from Vanderbilt University Medical Center. The DPC is led by Adam Resnick, Vincent Ferretti at University of Montreal, and Jackie Geneva from Seven Bridges. And you can see here, we've got a massive support network from the NIH through the Include project. And so it's really all hands on deck to make sure that these government resources and these really valuable data sets are being put to the best possible use and that we can get the most out of them. So what are the goals? We've heard a ton about this in the past 2 days, but the idea here is to understand these different co-occurring conditions in Down syndrome. We've discussed the fact that individuals with trisomy 21 experience a different disease spectrum, wherein they are protected from certain conditions like solid malignancies, hypertension, atherosclerosis, but they're predisposed to others. And we've talked a lot about Alzheimer's disease, autoimmunity, and cognitive deficits. And so we're really trying to put all these data together to understand this really complicated biological problem. So if you live with Down syndrome in the United States today, you have a 50% likelihood of being born with a congenital heart defect, a 10 to 20% lifetime likelihood of developing ASD, a highly increased risk of mortality from lung infections, as evidenced most recently and most prominently, I think, by COVID-19, a massive increase in likelihood of developing autoimmune conditions. We have here greater than 60%, I think, and I hope I convinced you that the number is likely much higher than that, probably upwards of 80 or 90%. The median age of Alzheimer's onset is 53 years old, and unfortunately, the median age of death is still 58 years old. And so while we've made great strides in the past 40 years in improving the treatment of these various co-occurring conditions and extending that lifespan and that quality of life, we have a long way to go. And that is the mission of the Include project. So one thing that we are very clear on is that diversity equals discovery. So everyone with Down syndrome is unique, and we need to view them all as individuals. That is where the greatest power lies. But in addition to that, a precision medicine approach is really the only path forward. There's not going to be one size fits all treatment. We need to take everyone as an individual, understand their needs, and design their treatment plans accordingly. So as we've talked quite a bit about what drives this phenotypic variation, I would argue we still don't know the answer to this. I don't think it's any one of these things. I think it's probably all of these things. So we know that everyone has an extra copy of chromosome 21. Some people have that in every cell in their body. Our self-advocates here do not have it in every cell in their body. So understanding what's common, what's not common, what are the genetic variants that contribute to these differential presentations, epigenetic mechanisms, the different endotypes. We've talked a lot about things that are not DNA centric. I think you guys have heard about what we call the central dogma probably a half a dozen times now. So there's differences in DNA, there's differences in RNA, there's differences in protein, there's differences in metabolites, and as we've talked quite a bit about, there's differences in the immune profiles of these individuals. What are the contributions of other factors? Environment, race and ethnicity, lifestyle, socioeconomic status, cultural factors. We need an orchestrated effort to enable testing of alternative hypotheses quickly and efficiently. And the only way to do this, again, is to aggregate data, because any one study simply doesn't have the resources and the power to drive these. So why do we want a data hub and a portal? In large part because it's the 21st century. So, the old way of doing things is, we sit in our silos and we collect data and we synthesize new ideas, and maybe we write them into a paper that a few of our friends read. And that's kind of the end of the day. And that's the old way of doing things. The new way of doing things is to deposit our data into open access databases where everyone can read everything that we've written, where everyone can see everything that we have done. And in a couple of really powerful instances, which anyone with any cancer biology research experience will know, see BioPortal or the Cancer Cell Line Encyclopedia. These are massive efforts that have been undertaken in the past 20 years or so to make data that has been generated with NIH dollars accessible and really interactional with the population. You can go in to see BioPortal now, click things in, you can learn about different genes that are altered in different types of cancer. Really powerful, very simple to use, and freely available, not just to researchers, but to the public. And these are the model that we're using to generate the Include Data Coordinating Center. Great. So this all started back in 2018, really, with the launch of Include. And then it's taken a while, I think, for us to kind of find our footing and for Include to know what all it wanted to do and how it wanted to realize its goals, which are very ambitious. If you have not checked out the Include website, you should. The stated goal is no less than to improve the quality of life for everyone with Down syndrome. And so by late 2019, we saw the first RFA come out where they said, hey, who wants to do this? What are your ideas to build this data coordinating center? We submitted our proposal in early 2020, and just with the government pace, we got our notice of award about 8 months later in September, and we started in earnest. It took us a little over a year to launch the DCC website, which you can go and access now, Include DCC. Google that and it'll pull it up. It's the very top thing. I'm not sure what the actual website is. I'm sorry. I have it bookmarked at the top of my browser. And then we have been loading in data sets that take us up to today. So when we launched, we had 5 studies. So we had the DS Connect registry helped to form the backbone of this, and this is mostly clinical and demographic data. Our Human Trisome Project data from the Linda Crnic Institute has clinical and demographic data, but as I described yesterday, genomics, transcriptomics, proteomics, metabolomics, immune profiling, so on. And then we had 3 studies from Kids First and Include centered on genomics and clinical and demographic data from individuals with Down syndrome and heart defects and pediatric leukemias. And so that was kind of our first 5 data sets that really formed the foundation, and we were able to move off and start running. In October 22, we added 2 more, ABCDS, which is clinical and demographic data, and then work from Dr. Kaur's group at the Benaroya, clinical demographic transcriptomic data. And then just recently, in this month, we added 4 more data sets, 1 from CHOP, 1 from USC, 1 from Geisinger, and we added another set from the University of Colorado Anschutz Medical Campus where we are. So we're constantly trying to grow this, and we're working to develop new intake forms and things like that so that we can work with anyone that's generated any usable data to the rest of the community, get those data put in a meaningful way such that we can aggregate them and hopefully use them for some higher powered studies. Great. So within 18 months of data, of launch, we had put up more than 75 terabytes of unpublished data. So we have a ton of published stuff that we have worked to pull in and portalize. There's still more for us to pull down that exists in some of the other NIH sponsored websites. We have more than 8,400 harmonized clinical profiles. So these are clinical dictionaries that they say the same thing. Anyone that's ever done clinical research knows this is an absolute rat's nest. Not everyone calls things the same things. Even if we use things like ICD-10 codes, you can have half a dozen ICD-10 codes that refer to the exact same condition, which can make mining these data really, really difficult. And so a lot of effort's gone into making sure that all 8,400 of these clinical profiles are in the same language and can talk to one another. We have 4,000 genomes. This is crazy. This is a lot of data, and there's so much that we're going to be able to mine from this. Again, we have a long way to go, but 4,000 full genomes is a really wonderful start. 1,000 plus transcriptomes. This is going to move very rapidly now that transcriptome profiling is much less expensive. And 400 plus proteomes and metabolomes. And again, we are working diligently to up the numbers of all of these things. Sorry, I have something on here. Great. So there's a set of principles that guide the way that we are bringing these data in, and I've kind of touched on these a little bit. There's a set of principles called FAIR, and what this means is that the data are findable, and so we're talking about automation here. This is the ability for people to write code to be able to find the kind of data that they're looking for. This is more difficult than it sounds, right? You can write the code, but again, if things are in slightly different iterations, slightly different languages, it's going to be very difficult for that search to return those things. So we want both people to kind of manually be able to find this, but we need the computers to be able to do it. As those numbers go up, it becomes, mathematically impossible for a human to mine these individual data sets. Accessible, so limitations on the use of data and protocols for querying and copying data made explicit to both humans and machines. So again, we want this to be accessible, but there have to be rules on how we're using this. And so it's important to govern those things carefully. Interoperable, I touched on this a little bit earlier. So the metadata, so describing the studies, needs to be standardized, again, controlled vocabularies, and have references to other metadata and be machine actionable. Which again, this all means that we just need these data to talk to each other in a way that's coherent to people and to machines. And then to be reusable, right? Why should we put a bunch of money into a study, we publish it once, and then it's gone? We need to have enough data that anyone can reuse these data to supplement, to augment their own studies moving forward. So this is a massive cultural shift that I would say has probably been going on for the past 20 years, if I'm being generous. In the biological sciences. And so this is kind of the old model of doing things. So, we start with taxpayers. Everyone in this room gives their money to the government through the IRS. There's some money allocated to Congress who decides what and how much is going to go to NIH. And then the NIH is going to decide which researchers to fund for which research, how and how much. This goes to data, which this is intentionally a black box because this has been the case for years, that you couldn't necessarily see how people arrived at the conclusions that they did. And at the end of the day, this led to a paper, which this is the ultimate carrot. So if you're an academic scientist, publications are currency. That's why this says publish or perish. You don't publish papers, you lose your job, you close down your lab. Unfortunately, that's a horrific incentive structure for making reproducible science, right? So More than a few postdocs have lost their careers to unpublished data, graduate students as well. And unfortunately, one of the other consequences of this that it's led to is sensationalism and grandiose claims that can't be reproduced, and we can't really see what the underlying data for those things are. And so this is where full transparency is absolutely critical. And so the new model, it still functions in the same way at the front end, right? So we still have money that's appropriated to Congress. NIH is still making the decisions, again, with the academic community on what are the most valuable studies and which are the most likely to succeed. But we break that paradigm here at the point that now data are going to be freely available and hopefully, again, interoperable and very usable at an early stage. And I want you to note that this is now flipped, that the data is coming out in open access before the papers. And this is actually a new NIH mandate that doesn't just apply to include. As of a couple of months ago, I'm not sure the exact date, but basically all of the grants that are funded moving forward in the NIH portfolio, you have to publish all the data that is generated with NIH funding by the end of the grant, whether you've published a paper or not. And so they're really putting their money where their mouth is across the board to say, when we pay for these things, that's a contract between the US government and those scientists that's meant to benefit the American people, right? And so when those data are generated, they are there for the American people. They're not there for one group to hoard and to use as they see fit. And so again, the hope here is that this is going to accelerate discovery by making these things accessible. So how would open science accelerate research that benefits people with Down syndrome? How would Down syndrome researchers benefit from this? And how does the Include DCC facilitate this open science? So I've mentioned sample size a couple of times, and sample size is everything. So anyone that's studying genetics knows that sample size needs to be really, really large because effect sizes, so the amount of contribution of any genetic change is often very small. When we look at some of these other, big omics studies, we can get away with fewer samples sometimes because the effect size is larger. But at the end of the day, getting enough individuals with a condition, in the case of Down syndrome, getting enough individuals with any co-occurring condition that we can do the appropriate comparisons, this can be very, very difficult. When we look at the top 3 or 4 co-occurring conditions, we may have 50 or 60% of the population. It drops off pretty drastically after that, right? So building a cohort for a co-occurring condition that happens maybe 3% of the population with Down syndrome can be very, very difficult for any one group to properly power. And just as an illustration of this, if you look at the population here, where we look at these different colored people, 14% of these men are green, right? And you take a subsample of this for the analysis, and if you don't have enough power, when you randomly select, you get no green men. And the conclusion there would be that there are no green men. Well, that's not true, right? We just didn't sample deeply enough. And so this is where bringing these studies together will allow us to put a much larger basket of the population, and then our subsampling gets greater, hopefully our power gets greater, and we're more informed in the conclusions that we draw from those studies. Enabling the study of underrepresented populations, right? Despite, I think, diligent efforts by many of the researchers in this room, we still have a pretty white upper middle class cohort across the board. And that's just, by dint of being in centers that have large academic medical research centers and people that have the time and the resources to participate in research has really limited our ability to diversify. And so this is obviously an area of emphasis, but again, it's one where the more we can bring groups together, the lower that bar is for us and the easier for us to draw some of these conclusions, right? And the types of questions, are there conditions that are more common, more severe in some groups? If so, why is this, right? Is that genetic? Is it socioeconomic? Is it lifestyle, cultural factors? An example at the bottom here, the epidemiological data indicate that there's a shorter lifespan for African Americans with Down syndrome. Why is that? What are we missing there? What can we do to change that? And Hispanics are the fastest growing segment of people with Down syndrome in the population, right? So we need to understand the differences in needs for these different groups. And in order to get there, again, we need a lot more data to properly power these analyses. So one of the goals of Include is to define the natural history of Down syndrome. But by bringing all of these things together, we're hoping that we can start to understand a more definitive epidemiology of the co-occurring condition across the lifespan, right? So perhaps someone with Down syndrome is born with a congenital heart defect, then they live a long life. A lot of stuff is going to happen. A lot of different co-occurring conditions are going to come and they're going to go in this population, right? And at the end of the day, they're going to have a higher risk of Alzheimer's disease, right? What are the rates of those co-occurring conditions, right? Are there sequences or codependencies? One thing we've talked about is that once you have one autoimmune condition, you're far more likely to have another. That's something that we've established by having enough data on autoimmunity in the general population, right? To make that kind of a rock solid principle. We need more data on the population with Down syndrome, population with Down syndrome to understand whether or not these same relationships, different relationships exist among the co-occurring conditions. And then are there developmental and clinical subgroups within Down syndrome? These are just a few of the questions that we hope to be able to answer by aggregating all of these data. Again, assessing reproducibility. One of the other things that can happen if you don't have a lot of data is you get one or two outliers that drives your result, right? And then something becomes gospel that may not actually be representative. We need really large studies for a lot of things in order to fully conclude what's happening, right? And so, I mean, here's just a wonderful example of this, right? This guy's got a large error bar. So what you can't see here, and one thing thankfully we've changed, we don't plot data this way anymore. We show all the data points. But this guy would say there's a lot of variability in that group, right? And so we cannot trust that this height is actually the representative height because the variance is represented by this bar, right? So we really need to understand what those differences look like. Exploring data before experimentation. So running experiments, running clinical trials, even clinical studies that are more observational. It's a lot of time and a lot of effort to pull these things together. The amount of work that goes into setting these things up administratively is mind-boggling. And so being able to parse some of these large data sets to more fully refine our hypotheses before we put a lot of time and money into them is going to save time and money in the long run. Finding biospecimens. For those of us that are interested in looking at molecular phenotypes like the ones that we've been discussing, we have a virtual biorepository. So what this means is instead of having all of our samples sitting in a freezer at one place, which does happen sometimes, everyone can collect their samples from their studies, they can store them, but you can build a study across sites. So now you can go in, if you're looking for X number of people with, say, celiac disease, and there's not enough at your site, but there's some at CHOP, and there's some at Crnic, and there's some at UCSD, you can build that cohort, you can request those samples, and you can pull them and have them sent to you, and you can perform your experiments. And so it really cuts down on the barriers to doing some of these types of experiments. Finding and connecting with collaborators. This is still something that is relatively difficult to do. We are a small community as far as research goes, but knowing what everyone is doing and finding where those areas of synergy are, it can be difficult, but it's really powerful, right? So these two guys trying to make the wheel, we got two halves, but their backs are turned to each other, right? So if we're not sharing the data, if we're not talking, if we don't know what everyone's doing, we may not see the whole picture, not to mention duplication of efforts, which is an unfortunate consequence of siloing and data hoarding, is not letting everyone know what we're doing. So another mission, outreach, engagement, and education. So here's just a sample from 2022 of all the different outreach efforts that the Include DCC has done. We are trying to take this to the community. So we talked to DISMIG, we talked to NDSC. There was a presentation to the Senate. We actually have regular meetings now for all of the investigators who are funded directly by Include. We're meeting a couple of times a year and trying to share these ideas and share these data. And then we've been doing more specific outreach. So going to SACNES, ASHG, Kids First, we're really trying to be out in the community and conveying this message to people, letting them know what we're trying to do and answer questions. I mean, there are still some people that are a bit skeptical of turning their data over to a bunch of people they don't know. And we're working on assuaging that fear and letting people know that we're just trying to put this together and make it the best use case possible. So we have a quarterly newsletter. We have 300 subscribers and counting. Again, anyone can sign up for this. An investigator registry, which is updated twice a year with grants and publications. If you want to know if there's anyone at your local university doing Down syndrome research that's funded through the Include project, this is all live right now. Again, you can go and search this. You can see every investigator that has received any funding, and you can see the grants and the publications that have arisen from those grants are all listed on the web page. We do biannual webinars, which again, open to the public. Portions of these are closed, but most of it's open to the public. And then we have office hours. And again, people can join and ask questions of these different settings. So one other thing we're really interested in is the data science for diverse scholars in Down syndrome research, which is a mouthful, so we just call it the DS3. And so this is a training event, and this is something that I'm very passionate about, which is we need to start training the next generation of Down syndrome researchers. I think for the first time, probably ever, this is a viable career path for young research scientists. It's been difficult to justify training people specifically in this when we couldn't guarantee there would be a funding stream for them to establish an independent research career. That's no longer the case thanks to the Include program. And so now we need to build this pipeline out. And we have a chance, because there is no pipeline right now, to make sure from the get-go that this is a diverse group of individuals. And so we're teaching students how to perform fair research in the Data Hub and beyond, training students in the skills necessary for big data management, analysis, and visualization, which I think we can all appreciate. Big data is the way of the future, not just for science, but for everything. So even for my molecular students, they're learning how to do all this big data analysis because it's the way that everything is moving. And then to empower diverse trainees with professional skills necessary for career advancement. So what this is a 2-week course that's going on right now in Boulder, Colorado. It started on Monday. It will wrap up next Friday. We fly in students and postdoctoral fellows from all over the country. Here's our class of 2022. You can see this was a little bit smaller of a class because it was our first year and we needed to make sure that we could do the things we said we could do. But once we iron out some of the kinks, we were able to recruit a much larger class this year. So these individuals are in a coding class right now in Boulder, learning from a bunch of really talented bioinformaticians. And then they get a bunch of expertise. So this is all anchored on the Crnic Institute. Many of our instructors drive up from Denver to Boulder and give lessons on data science, but also the biology of Down syndrome. And then we fly people in from all over the country. I don't know where it really went, but Anita Bhattacharya gave a talk yesterday. And so we've been trying to include as many diverse voices as we can. I don't know the rest of the program. So our goals and milestones for the next 12 months is to surpass 20 studies in the Data Hub. We're currently at 11. So this is ambitious, but we think we can get there. Surpass 800 registered users. We have 432. I haven't checked these since I sent this to Colleen the other day. These numbers have probably gone up a little bit. 10,000 unique research participants. We're at 8,400. 5,000 whole genomes. We're at about 4,000. 2,000 transcriptomes. This one I'm pretty confident we're going to beat this year because we have another 700 at the Crnic Institute that we're processing right now. And then transition from data collection and sharing to data analysis. And so one thing I didn't talk a lot about because we're not fully there yet, but this whole data hub where all these data live, where you can kind of click through and see, this is all directly portable to a cloud interface where you can do computing on multiple processors on these really large data sets at really no cost to you. In fact, Include, for the investigators in the audience who aren't aware, is granting credits for computational time in the cloud because they really want people to come in and utilize these data. And we're hoping we'll have the first couple of scientific discoveries that have been enabled wholly by this hub in the next year. Goals and milestones, extend and expand DS3. We're very excited about this, and we hope that we can get more and more individuals in and really build out this pipeline of young investigators who are passionate about helping people with Down syndrome. Streamline the integration of biorepositories. This is another rat's nest, the way that people keep their information on biorepositories, but we are making great strides. We have some wonderful colleagues that have big biorepositories that have been working very diligently with us to figure out how best to harmonize these. Integration and analysis of imaging data. So this is something we haven't really considered yet. We've been looking mostly at things that are much easier to integrate, large spreadsheets. Imaging data tend to be very large. And so we're working on figuring out how best to share these and make them searchable and usable, excuse me, for analyses. And then demonstrate use cases for interoperability with other data resources, right? So this is something that we've been waiting until we had our system fully set up, but we'd like our data to be able to talk to other data sets outside of the Include DCC. And then something I'm personally very excited about is to develop a pilot researcher portal for the sharing and analysis of experimental models of Down syndrome. So everything in the DCC now, these are primary human samples, but as we've heard, those things can really be buoyed by experiments using cells or animal models of Down syndrome, and that's something that we had contemplated. We just got a supplemental grant to this DCC grant to work this up in the next year and start to include many of our colleagues who are working on those systems. And yeah, that's my last slide with that. I don't know if I have time for a question or not, but if we do, I'm happy to take it.